story that I'd never heard before. My mom was telling me that my dad had told her when he first met her that he was going to go to Japan. He had no plans to go out with my mom or anything. He just met her. And he said, you know, the reason was that they were low maintenance. They don't need all these modern things. He hasn't been right. It has changed. And actually, when I would have been 2 years
on a lighter note, how was your son?
He lived there for 3 years. He got the taste of, you know, what didn't like. Anyway, he kept telling his friends, well, you know, I can learn Japanese. You know, arms are as low maintenance as you can get.
I have done nothing recently.
Makes you feel
Has anybody talked to you?
Went all very, you know, down to earth, from a long line of farmers.
People more involved in the artwork side of it.
I didn't get upset when he was like a background yeah, I would be pretty ticked off.
Can I have everyone's attention? Todd said no, but I think the rest of you said yes. Welcome. We just figured out this is the 8th iteration of the GES Colloquium. Let's give ourselves a hand. It's pretty neat. It predates the Oecd by 1 year, and then every year that we had an Oecd cohort, we've run this. And I think what's really fantastic is that we've been able to continue this Colloquium series beyond the life of the Oecd. And that's worked really well. And it's because you all keep showing up, and many of you agree to speak, and we keep having interesting conversations. I also want to give a shout out to Patty, who's filming this today. If anybody logs in to watch this, I'm sorry. I go and talk in different places. I'm really proud to give our website as a resource because there are an amazing set of talks available online, and that really represents the diversity of perspectives and expertise and thoughts and values that our center brings together around these conversations. So, so keep coming and keep telling people, inviting people to come with you. Know that resource is online. Also want to thank Sharon Stauffer, who's not in the room today because of health issues, but she's been an integral part of making this colloquium continue to happen in terms of logistics for outside speakers. We've been fortunate that the GES Center has allocated money every year to provide funding for some external speakers. And I know that if you think about what this colloquium is like, just the infusion of some outside people and the chance to take them to lunch and meet with them and have them learn about us and we learn about their work, that enriches our whole community. And I want to urge all of you, the other innovation that Patty's come up with, which is really neat, is we've been talking about how when we spend money to bring people here, we want to make sure that you all have a chance to meet with them. And so on the announcements for speakers, if they're coming in from out of town, there's a little box that you can click that says, I'm interested in meeting with this person. You don't have to be an expert. You don't have to be thinking that you want to do a one-on-one meeting where you grill them about their research and have some intimidating conversation. This can be about, let me get to know you. Let me find out what you're like as a person. Let me think through how my work and ideas fit with yours. I mean, this is a great networking opportunity. It's also a chance for us to leave an impression on people who visit here about the breadth of work that happens here. Most speakers who come here are really excited to spend time meeting students and meeting other faculty. It's not an imposition. They've already been imposed upon to travel here. Let's make their time worthwhile. So do notice when speakers are coming. You have to think about that a little more than a day ahead of time. But again, Patty's innovations in the way that we announce these colloquia is that you see who are the upcoming speakers, and you can let us know if you're interested in meeting with them. Sharon has graciously agreed to coordinate our speaker schedules, and that's a big job. But it means that all of us can get together with people. I'm never able to go to lunch right after this because I teach right after this, but we're always interested in having students and faculty volunteer to take our outside speakers to lunch. just a couple other logistical notes before we go around the room and give our updates. So for students in the room, if you would be so kind to sign up for GES Colloquium as a 1-credit class, it is GES 591-002. We are in the process of making this a more formal, ongoing class that's pass, no pass, whatever that is. But that paperwork is in process. So this semester, it still exists as a 591 class, 1 credit for a grade. And I can discuss with you individually how the grade is determined. About that. Very rigorous. And we piloted the GES discussion last semester. There was confusion and difficulty in getting it into the schedule. It didn't appear. I found out that if I wanted to try to get it into the schedule, I was going to ruffle some feathers for strange bureaucratic reasons. So that mini discussion project is on hold, but we'll plan to offer it in the spring as a pass, no pass class. It'll be a discussion on Thursdays that will pair with a colloquium, so I'll do that again. Any questions about how this works? Questions about the semester? Okay, great. Well, the main reason to come today is eating and also catching up on all the interesting stuff that folks are doing. So we'll go around the room. We have 45 minutes, so each person can take a couple minutes to talk about especially what have you done this summer related to GES. What are some of your plans over the coming academic year? So that's what we'll do today. Megan?
Oh.
I see that you had just taken a bite, so I thought I'd call you instead of Nora. We'll just go around the room.
So I'm Megan Sear. I'm in the biology department, finishing up my PhD this semester in working with Dr. John Guzman, our new postdoc, Kelsey Horne, next to me. And so I work on all things wild mice, particularly invasive ones on islands, and particularly thoughts of how to get rid of them using gene drives and other technologies. This semester, besides trying to finish up, both Katie and I are helping out with Dr. John Godwin's class on genetic engineering for human health and the environment. Something like that. Something like that. Which he's actually doing that right now. So in 2 weeks, I'll be taking the students. We have about 8 excellent students who will be coming here to also be participating in Colloquium, and then kind of having a discussion as well, so when they are able to attend. Beyond that, I think that's about it. This summer, I had a pretty busy summer in a tick-infested world building enclosures, hence the bruises. But I also adopted a dog. I named her Molecule, Miss Molly. So that's about it.
Hi, I'm Katie Barnhill-Billing. I'm no longer a PhD student. Yeah, so that was what I did with my summer. I started August 1st in biological sciences, working with John's team. I think Royson's here. Yeah, there was an exciting dynamic phase with the Safe Genes project, and I'll be working as project coordinator now, and doing some of the stakeholder engagement with Jason. So that's been an interesting transition, and our lab meetings working on mice issues are very different than our research team meetings around social science. But they've been very welcoming. It's been a great start. I taught math history too. I taught Intro to STS online this summer, which was a really engaging experience and not at all what I expected with online teaching. It was really positive and really, it was a great experience. Yeah, and also my daughter is 21 months old today. We spent a lot of the summer teaching her, or she's teaching herself how to swim. So that was really, she has a little pella jumper, don't worry. But it was really fun, and it's just really fun to see the world through those tiny little eyes, because it's a very different perspective from ours, I guess. Yeah, so, oh, the other thing that we're doing this semester, or this academic year, I guess, is following some conversations we've had at different events and building off of some dialogue we've had in different places. Nora and I are convening a sort of online space for women in the GES Center and beyond, women who are interested in genetic engineering, engaging the way gender works in this space, both in terms of content and in terms of professionalization of how we handle working as women in spaces like this. So keep an eye out for some of y'all here, keep an eye out for an email. We're really interested in keeping it open beyond the GES Center as well, but we're starting here. And we're really interested in just creating a safe and supportive environment and using that network to sort of, I don't know, help us be us.
Great. My name is Adam Kokotovich. I'm a postdoc working with Jason. One fun thing I did this summer, I was able to take part in a report that the IUCN, a large international conservation organization, was doing, looking at the ways that synthetic biology may help or harm conservation efforts at a global level. So we went down to Brazil and had a writing workshop where we worked on advising a variety of chapters. I was working on a chapter having to do with evidence and the ways that we understand how values inform how evidence is created and the relevance of that for those discussions. The main project I'm going to be working on kind of going forth has to do with spotted winged Drosophila, the invasive berry pest. As a social scientist, you can ask me questions about that and I won't be able to answer too many of them. But I'm going to be working on some of the values-laden and social sciencey questions around risk assessment having to do with different techniques for addressing this pest. So we're going to be doing some problem formulation work around the 5 different techniques that exist for addressing it, including some of those that involve genetic engineering. So that'll take up a large portion of my time this coming year, in addition to helping with other kind of lab-based projects like the engineered American chestnut and things like that. So.
Hello everyone, my name is Dalton George. I'm a second semester PhD student in forestry and environmental resources. This summer I spent a lot of time working with Jason on a couple of projects, including G-Bird and the American Chestnut project, cranking out a workshop report and landscape analysis report. So kind of jumped in with both feet on those, which is a really good experience so far. I've also been working with Todd on his DIY work. Really excited to get more into that this next coming semester and in the spring. Taking a couple of methods courses this fall and spring, and trying to find a nice foothold for my dissertation work.
All right, I'm Andrew Hardwick. I'm a senior with a focus in environmental policy. I'm here to learn more about GES. I've been here since last semester in the spring, and I'm excited to work this semester in your class on natural resource issues and policies and how we can apply things we learned in this colloquium to that class. Over the summer, I got to work at RTI in their Environmental Technology and Energy Economics department with a few different EPA projects, and that was a lot of fun.
I'm Darby Orcutt. I'm with libraries. And the big announcement, I guess, is just that I'm now your sole official libraries liaison to the center, the cluster. My colleague, Mohan Raheshmari, who many of you may have worked with, has departed the university, sadly. Again, though, that role of library liaison, continue to do what you do, but certainly feel free to reach out to me or to loop me in as you may need me. Thank you.
Hi, I'm Jennifer Cruz, and I spent the summer reacclimating to North Carolina. Having been gone last spring, I missed all of you, by the way, doing the Fulbright in Canada, and that was a really great experience, working on the transgenic salmon issue, the differences between Canada and the U.S. Really interesting. So. Yeah, over the summer I've been working on a lot of small things for the center, and they've been adding up to a full-time job, but none of which are that significant to talk about yet. So yeah, but what I did 2 days ago, I dropped my daughter off at NYU for her first year of college in the middle of New York City. And so I'm a little out of it today because I'm like, is she okay? I don't have a text yet. So if you're listening, honey, I hope you're okay. I know she's not listening. She's English and history major, so yeah. Anyway, but if you're interested in any of the stuff I've been doing, little stuff for the center, mainly program development and connections and project building. So there we have it.
I'm Matthew Booker. I'm in the history department, and I also direct the STS program, Science, Technology, and Society, which is an undergraduate program that Katie helped us with a lot this summer. I guess I'll stick to 3 things. 2 of them are requests. I'm teaching U.S. agricultural history this fall. 90 students are enrolled. It's the second time, I think, in 40 years that the course has been taught at NC State, and the first time that I know of was in 2016 when I taught it. The best thing about that class, according to the students, to a survey I did, is the guest speakers. Overwhelmingly, that was the thing they were most interested in. Which is right. which is a compliment to the guest speakers, including Fred, who's coming in. So that's happening, and you are all welcome. It's a large room. We meet Monday, Wednesday, Friday from 9:35 a.m. to 10:25 in 321 Riddick Hall. And there's probably seats at the back. Come on in. Don't play with your phone. And also with me in the room is Kelsey Horne, my teaching assistant, and a brand new graduate student in the public history program. Kelsey's from Mississippi, and she's been adding a lot to that course, both from her own experience and from her interests. 2 requests. First, it looks like I'm going to apply for a Global Engagement Institute. Yes, and I've asked a couple of you for specific help on that. It's in East Africa, and there's a chance for me to visit a couple of different institutions, or perhaps one institution over spring break. I'd like your feedback, your suggestions about how I might take advantage of that, thinking about GES as a frame. And then finally, I feel like there's one more thing I really need your help with. Yes, there is. Yes, there is. The Buchdahl, the annual Buchdahl lecture, which is the STS's kind of prime event, will probably be in spring again, and I'd like your suggestions. I'm soliciting good ideas for voices around food and agriculture. I think those would be especially interesting. I look forward to that from you. Oh, and then the last comment would be, sorry, GES, the Archive of Agriculture, Genetic Engineering and Society is humming along, thanks to Sharon, especially thanks to Sharon, but we expect more interviews this fall.
I think we have our next one in a week and a half, I think.
It's been fast.
We had lots of undergrads, well, 3. Especially when we lost, one of the supervising grad students for one of the projects. He was away at Monsanto, so, me and the rest of the lab picked up supervising his student. We had a couple of students for the BIT summer program. I don't think we got any papers out, but definitely working on wrapping up that one last thing that needs to be done, and submitted a grant. So it's been a busy summer.
I'm Marcella Lorenzen, and I would say that I'm still in shock that the summer's over. It just seemed like it zipped by faster than ever, probably because we had 3 undergrads on top of all of our other undergrads, because these were 3 summer, what do they call that, research experience for undergrads students. So they were 2 NSF funded and 1 USDA funded. But 2 of them were working on Medea, which is a selfish gene that says inherit me or you die. And those students did a great job of sifting through all of our transcriptomic data and digging down and trying to figure out what goes on in these critters that are dying. So how does Medea get an advantage? And we're hoping that we can maybe publish on that in the next few months, along with a bunch of other papers.
Should we skip back to Michael and
well, so yeah, I'm Michael Bella. I'm a PhD student in the Biomathematics PhD program, working with Fred and Alan Lloyd. I guess I'm going to be working to actually write up results that I've gotten throughout my time here so that I'll be able to graduate sometime next year. one of the projects and something I did this summer was visit our collaborators, Tony James out at UC Irvine. They're doing really exciting work with Chain Drive Anopheles mosquitoes that can't transmit malaria. So we're helping them analyze their data, that kind of thing. That's about it.
Hi, I'm Kevin Gross. I'm in the biomathematics program. I've spent my recent summers in a somewhat new collaboration with a friend of mine on the science of science, which involves a lot of navel gazing, it must be said. I've been thinking more recently about how institutions distribute funds to advance and promote and fund science, and how experts decide upon what science to fund when they disagree on things like what constitutes evidence.
Hello everyone, my name is Royden Saw and I'm a visiting scholar here at NCSU, hosted by Fred, and I coordinate the Genetic Biocontrol of Invasive Rodents program, which is the nonprofit group that is really looking at whether genetically engineered rodents can or should be used in eradication projects. And I guess the main thing that I was able to do is attend a staff retreat with my boss, Carl, who lives and works out of the Galapagos. And so was there for a couple of weeks on a small island called Floriana, which has 140 residents and a bunch of tourism, and trying to figure out how they're going to use conventional pesticides to conduct a multi-species invasive rodent, invasive animal eradication. So it really lent me the ability to see why new tools are used, so it opened my eyes quite a bit. And of course it was the Galapagos. So, good times. Thanks y'all.
I'm Mitzi Porras. I finished my PhD at Penn State. I was working with Consuelo de Moraes, and now I joined the lab of Max Scott, so I'm postdoc in his lab. We are working trying to develop a lethal strain of aphid or eggplant hopper. So that's my task.
Dr. Berger kind of already introduced me, but I'm Kelsey Horne. I'm a new MA and public history student.
Welcome aboard.
I'm Jen Baltzegar. I'm a PhD student in the genetics program in Fred's lab. And this summer, I was kind of cranking away, trying to get stuff done to maybe, hopefully, one day graduate. So nothing really specific to talk about. TAing for the first time at NC State this semester. First class is this afternoon, so we'll see how that goes. Genetics 311 problem session. I have 2 sections of that.
Hi everyone, I am Esteban Nieves, originally from Mexico City. I'm on my 2nd year as part of the Master in Microbial Biotechnology. I guess that this semester I'm going to be working with small projects with agricultural companies. Last summer I performed an internship at Indigo Agriculture, a startup that is located in Boston, but they have offices here on RTP2, and I perform, well, it's a microbiome into plants company, nothing with genetic engineering, yet agriculture.
Hi everybody, I'm Nicole Guzman. I'm a PhD student in the entomology department working in Marcel Lorenzen's lab. Right before the summer, I finished my prelims, yay! And then over summer, I submitted a USDA grant that I think has a good chance of being funded, so I'm crossing my fingers there. And then I TA'd for half the summer, and then the rest was mostly just like life events. So my little brothers graduated from undergrad, so I was able to fly home for their graduation, so that was fun. They're already making more money than me. Should have done physics or computer science. I went wrong there. My grandmother passed away, but I was able to see her at the beginning of spring, so kind of bittersweet there. And then I went to Hawaii last week and was there for Hurricane Lane. I'm here and I survived, and that still didn't stop my now fiance from proposing. And now I have to get back into the swing of things, and today I haven't gotten anything done. Hopefully tomorrow will be a new day. Yeah.
I'm Paul Enriquez, a PhD student in structural molecular biochemistry. I didn't have as interesting things happen to me, like going to Canada or Galapagos, which I'm really jealous about, or get married. But I've just been working on a few papers, and this fall I'm going to be submitting some papers, one related to some GMO labeling stuff, and then the other one about certain restrictions on germline editing.
I'm Eli Hornstein. I'm a third-year PhD student in plant and microbial biology. We're really finally phenotyping a lot of them in a way that makes me confident that they're not doing what I might hope they would do. It's possible they're not doing anything at all. We'll find out later. So in the coming semester, we're pushing into making our next planned generation of transgenics, which have some modified versions of this and potentially some other proteins. And I'm also looking at typical graduate student things like doing my prelims and trying to make a paper out of the last one.
I'm Mike Jones. I'm just starting my 5th year in the Ag and Resource Economics department. This summer I passed my oral prelim, and then we were able to submit a paper that hopefully we'll hear some good news on. It's been out long enough that it's not immediate bad news, I guess, but we'll find out. Some cool thing happened actually stemming from the colloquium. So I gave a colloquium last year about public perceptions, potential consumer responses to gene-driven insects in agriculture. And someone from the EPA was here in attendance, named Caroline Ridley, and she actually asked me to give that talk, or an adaptation of that talk, to a biotechnology conference that she actually interfaces with folks in the USDA, EPA, it turns out even people in the Foreign Ag Service. And so that was, it's nice when people think what you're doing is cool. So, I got some good feedback from them. One of the more famous plant protection people had to listen to me for an hour, which is crazy. I stopped watching it. But, so that was cool. And then the rest of the summer, I watched my little girl grow up even more. She's 7 months old now. Crazy, just lightning speed, but really cool. And working on making progress on a lot of the recommendations that came out of the prelim. I think pushing some along pretty well. Presented at our national disciplinary conference in DC. Got a chance to interface with folks from the Foreign Ag Service about gene drive topics and some genetically modified insect topics in agriculture they hadn't really thought of before. I think I might have scraped them. And then trying to move forward. Oh, in this semester, one thing that's big for me is that I'm teaching for the first time, an environmental economics class, first time I've independently taught. And so I have a class of 50 undergrads who actually seem really enthusiastic and really responsive in class, and it's going to be a ton of time. Kudos, folks, that balance others. But I think it's going to be really rewarding.
I'm Fred Gould. On a personal level, my daughter left for college when she was 18, and she's now 37 and moving back to Raleigh with her family. And I have a grandson who's 2 years old, so that's going to be interesting. Professionally, I wanted to say I'm very proud of the fact that Jennifer and I and Zach published a paper in Science together that I think really put together the idea of GES, because we had a paper that Zach came up with the name for called Wicked
Evolution.
And the rest of it was something. Anyway, we forgot. But, you know, it really showed that.
It's tackling the socio-biological dilemma of pesticide resistance.
Yeah, that's why we didn't remember. In any event, that was really cool because, so I was proud of that. And also, a lot of good things have happened. So Jen and I got, and Alan got an NIH grant to continue our work with mosquitoes from mosquitoes, and that's a 5-year grant. And then we got this NRT, the National Research Trainee Grant. So that is a cool thing because it enables us to continue some of what we have going on here. And we have a lot of the co-PIs and everybody around here. But man, we really need input from you students in terms of giving us, helping us in focusing this thing and in recruiting new students, because you students are going to be the best advertisement we have for this thing. So we'll be giving a colloquium sometime in late October, but we may have other meetings because we really have to get on the ball in terms of advertising and in terms of getting things going.
Okay, I'm Keith Edmiston. I'm a professor in crop and soil sciences, and I'm also the extension cotton and hemp specialist. And so Marcella said she was glad summer's over. Unfortunately, summer's not over for us. we're harvesting hemp now, but cotton will be going on for a good while. So if y'all get really tired of teaching lab work and you want to go out one day and pick some cotton, just let me know. One of the things that and one of the things I've done this summer that would be related to genetic engineering is that in our program we evaluate germplasm for companies, and a lot of that in cotton, at least most of it is genetically engineered. Certainly not in hemp, although I had a meeting about a week ago, the first time that a company has expressed interest in genetically engineering hemp, which would be interesting to see.
I saw that article in Nature. There was a Nature article about that. I don't know if it's the same company, but it seems like a big deal now.
It'd be interesting to see how that's accepted, because in general the people that are really pro-hemp are sort of the other way. One of the things I have going right now that may be of interest to you is I teach a class, and like Matthew said, the best thing about this class is I don't teach much of it. It's all pretty much people like Scott Shore here, people that are smarter than me, as guest lecturers. So industry came to me and said they would like to see our graduates have more experience in regulatory science and regulatory affairs. So I had to reach out to industry to help fulfill that, because my experience with regulatory science is field trials. That's a very small part of all of it. And I realized when I work with companies and conduct research with them and meet with them that a lot of the times our discussions turn to regulatory science. We start throwing out these acronyms and things that I don't know anything about. And as I started teaching this class, I realized a lot of people that are in regulatory science and regulatory affairs did not plan to be there. Most of them didn't. Most of them were people like me that never envisioned that that's where they would end up. And with my graduate students that are in industry, even if they don't end up in regulatory science, they have to interact with the regulatory folks all the time. So I think it's it's good to get some experience and some knowledge of that. The class met one time so far. It's online and in face. Either way you want to take it. You take it either way, or you can sort of mix it up if you want. So if anybody does want to sign up, there is room. You've only missed one class, and it's offered on Mondays from 3 to 5:45. So I can take extra people in the class. Eli's taking the class. He was one of the best students we ever had, and the other one was Jennifer's.
Jace.
Oh.
Who's working for Genome Canada right now.
Right.
Hi, if you're out there, Jace. I'll just keep waving to the camera for everybody else.
It's
Hi,
pretty
Jace.
interesting in this class that you get people from wide disciplines, all the way from Jace to Eli and people from entomology and everything. So, thanks.
Go back first to Scott.
Okay, I'm Scott Shore. I have an adjunct appointment in plant and microbial biology, and I also work as a consultant for USAID and Gates-funded projects for agricultural biotechnology in developing countries, Asia and Africa. Primarily, I give a colloquium talking about some of that work. with cassava. And then I also work with a few other colleagues on gene drive and facilitating preparation of government regulatory people in Africa for gene drives. And so we've had various workshops that have taken place that I've participated in, and with the support and advice and comments from Todd and Fred this summer, I attended the SUBSTA, which is the subsidiary body on scientific, technical, and technological advice.
You survived.
As part of the COPMOP, the Convention on Biological Diversities, and the Cartagena Protocol. And they were discussing things that came out of the COPMOP of Cancun and are going before the COPMOP that will take place in Sharm el-Sheikh in Egypt this fall, where there's two main things of discussion on risk assessment of living modified organisms with specific inclusions with a parenthetical, which means there wasn't consensus on agreement to include it in the language on gene editing, but definitely including gene drive organisms and living modified fish in an overall risk assessment structure they're looking at. And then also on synthetic biology that's under the Convention on Biological Diversity and how to define that, which they still haven't done, but a lot of discussion on that. One that because of what I'm paid to consult on, I wasn't involved with, but was real interesting to listen to was the discussions on DSI, digital sequence information. So how that information is taken and used throughout the world and equitable sharing of that. And so there's lots of interesting things that are happening at that point. I don't know yet whether I'll be going to that. I don't know whether Todd's planning on attending. Okay. They've scheduled it to take place over the U.S. Thanksgiving. It's November 17th to 29th, so it'll be difficult. It's a 2-week meeting. But part of what we did was try and help our African colleagues understand and prepare for the whole COPMOP process in those meetings. So we held a workshop just a week or so ago in Nairobi with the help of one of our African colleagues who organized it, where we had some of the focal points for the Cartagena Protocol attend, and had Dave Obrakta, who used to be at the University of Maryland, give a introductory talk on gene drives where they got a chance to ask questions about it and understand it technically. We had our African supporting group, NEPAD, which is under the African Union, the new Partnership for African Development, also gave some talks there. So trying to get people engaged and prepared to be able to participate in that, and whatever they felt their institutions and governments, the positions they to take on gene drive, and not just sit back and listen, but actually participate in those discussions. So I think that was good. They're having a couple more meetings before November. What I'm preparing for is we're organizing a workshop where we're trying to have, instead of what's been called training workshops, it's a consultative workshop. We're going to work with a targeted group of government officials to develop programs for how they might approach risk assessments for gene drive organisms. And because of the work that the Target Malaria Project is already doing in Burkina Faso and Mali, although it's not on gene drive yet, that's where they expect to be submitting applications to first do it. We're going to have our first workshop there in October, where there will be a select group of people from the National Biosafety Regulatory Authorities and some malaria mosquito control people talking about how they might do risk assessments on gene drive. What would be the same as what they've done for other mosquito malaria control programs or other biocontrol programs, what might be different from gene drives, and how they might approach doing those assessments.
Thanks. And I think we've got about 6 people left and about 10 minutes, so just checking in on time. Please.
Hi everyone, I'm Sophie Cathario. I'm in food science and I'm an adjunct in plant and microbial biology. And I work with bacteria that cause vomiting, diarrhea, and worse. But actually we're looking at their adaptations, heavy metal resistance, antibiotic resistance, how they survive on surfaces in nature and so on. Thank you.
I'm Zach Brown.
I'm sorry, Patty. I mean, you should go next, Zach. Patty?
We have been pretty busy
up on the website. If anybody here is artistically inclined, we are accepting artist proposals for the artwork in the Age of Biotechnology, Shaping Our Genetic Futures, which is really a short title, and I don't know if it conveys what we're doing with that. So that we're going to be accepting artist submissions through, what is it, Todd? October 1st. October 1st, and he can talk more about that. But make sure that you're signed up for the newsletter. We make all of our colloquium announcements there. If you miss any colloquia, we're filming them and they'll be on our YouTube. Please subscribe to our YouTube because YouTube doesn't let you have certain access to certain features until you have enough subscribers. So if you're not yet subscribed, then please do. There's a link on our website and in our newsletters and everywhere else. And if there's anything that you want to share with the community or anything else, papers that you're publishing or anything like that, let me know and we will put it up there. Thanks.
So I'm Zach Brown. I'm an assistant professor in Ag and Resource Economics. I'm also part of the GES executive committee. Probably two pieces of GES information from the summer, just to keep it short. One is we had our graduate training capstone or graduate training workshop in June. And that was originally supposed to be a capstone for the IGERT, which many of you know was coming to an end, but also ended up expanding into something bigger in terms of looking at interdisciplinary graduate training. And so one thing I want to say to that is that many of you participated and helped in that meeting. So thank you again. I think we got a lot of really good feedback about the meeting. And all of the, we also, for those of you who were there, some of the talks were amazing. And we have almost all of them, at least the ones that are not from industry, I guess, are all streaming online. So I would, that's one of the important products. We're still going to do some more, try to do some information dissemination from the meeting and some of the lessons learned. But right now, there's a lot of the material from the meeting is just available online. So that was, I think, a rewarding experience. The second thing is that from another meeting that was years ago now, there was an OECD-sponsored meeting on international governance of gene drives, actually, that one of the projects that came out of that meeting was to work on a series of proceedings papers. So all the authors, all the presenters who participated in that meeting were under an obligation to produce some type of paper coming from it. And so we finally got all of those papers published as a special issue in BMC Proceedings. So that came out this summer. It's an open access issue, and for a proceedings issue, there's some really impressive, really nice work in there, both in terms of just summarizing an array of different countries' governance systems and how they would approach a technology like a gene drive. There's one paper on NEPAD specifically on how they would approach a gene drive. And then there's some other papers that are kind of bringing together risk assessment people and social scientists. There's one example with, actually Jason's the co-author on, of a nice kind of integrated paper that has very GES flavor to it. And all that's open access, and so I think I'm going to want to work with Patty this fall a little bit to try to disseminate that more. So in terms of going forward, this fall, the main thing that I want to be working on for GES is coordinating this NRT PhD program, and so part of that for me is going to be relocating myself physically for at least 1 or 2 days over to the GES Center so I can really focus on getting this, because this is going to, like Fred said, this is going to be a big lift to get this set up. So looking forward to it.
5 minutes left, 4 people.
Hi, I'm Todd. I'm a scholar in the GES Center. This summer I worked with Adam on the IUCN report, which we're anticipating 1,000 or so comments that will be coming in. We have to address 967 would be a deal number, and I win the pool of money. I've also been busy visiting community labs as part of a project that Dalton is helping me with, and we're almost done with the US ones. Next week I start on the European labs, but I've also been working on this art exhibit, the art's work in the age of biotechnology, shaping our genetic futures. So we have the open call out. If folks are interested, please come talk to me or Fred. We're also working with Molly Renda at the libraries on this. This will be an art exhibit in the fall of 2019, held at the Gregg Museum and also the library's exhibits. And this coming summer, if you're interested, we will be growing an artistic corn maze at the North Carolina Museum of Art. which they are for some reason going to let us dig up a portion of the soil on acreage. So it should be an interesting experience to put that together and watch that grow as well. And just maybe non-GS related, I'm excited in a few weeks I'm taking my first curling lesson.
Okay, so I'm Nora Hahn, and I teach anthropology. Okay, so early part of the summer, laying the groundwork for a new project, gendering GM, and getting that underway. Also thinking about internationalizing the GES Center, so I heard a lot of good stuff here, and maybe think about using this year to start laying groundwork for an international network of GES type centers. Maybe we can get some people invited via Skype. So all of that happened, and then I got good news on a book manuscript. So Oxford University Press gave me the verbal that they're going to take it, which of course last minute rewrites. Okay, but the really exciting thing, going back to what Zach said, this was actually the highlight of my career maybe. I wanted to get into popular publishing a little bit, so I had my first article on The Conversation. I don't know if you all know this.
Nice.
Right, so that article was about a gender-targeted program in Mexico that supports 25% of the Mexican population. And 2 days after the article went up, somebody from the transition team from the incoming Mexican president wrote us to say they're taking position papers, they're revisiting the program, would we submit one?
Wow.
It was thrilling. It was thrilling. It was thrilling. And so hopefully going to free a lot of women's time, because it's a conditional cash transfer program that takes up a lot of time.
Thank you.
I'm Jean Rustino. I'm Director of the Emerging Plant Disease and Global Food Security Cluster at NC State, and a plant pathologist in the plant path and entomology department. And the last 6 months I've been on a Fulbright Award in Italy, in Catania, University of Catania, where I worked on lake blight on potatoes and tomatoes. But I also did a lot of other things besides working on lake blight. I got invited to a Gates convening in Rwanda where we're visioning an African plant health science initiative. The recent call by the Gates Foundation was for disease surveillance and diagnostics, and they're funding these Grand Challenge grants, but they're going to put about $24 million into work on disease surveillance, and this includes pests and pathogens. So we had about 50 people in the room, and we kind of visioned what that initiative is going to look like. And I've been doing some phone calls with them on this initiative as well after the meeting. I also gave a talk at FAO. I see Alan Ruska is going to be here. Alan invited me to FAO in Rome, and I gave a webinar on emerging disease and global food security in Rome. And also, when I got back, we had a big International Congress of Plant Pathology in Boston where I led a keynote session on emerging disease and global food security. And I'm editing a book on the subject, and we've written a white paper that we're submitting to Science. It's a multi-authored paper, about 15 co-authors on things that need to be done to move the science forward in this area. And we have kind of bullet points, and it's a policy type paper. And then the book will be coming out in the spring as well. I got a phone call after the sessions up in Boston, and there's a freelance reporter that's going to do an article on emerging diseases. She's submitting it, she's planning on submitting it to Science, and she's also, she writes freelance for Science, but she's also doing stuff in the press, at New York Times. So I talked to her quite a bit about emerging diseases and how they threaten food crops. The session we ran at ICPP was called How Crop Diseases Affect Your Breakfast. And we talked about breakfast breeding, we talked about Panama disease on bananas, we talked about lake blight on potatoes and coffee rust, kind of breakfast foods. We had 300 people in the room. They took the Ubers and subway to get there because it was at the Harvard Science Museum. And so we got a nice publicity on that, and we have on my website, you can see they videotaped it, so we have a live streaming of that. And in terms of genetic engineering, I have a student in my lab now from Nairobi, Kenya, and she's been working on looking at pathogen populations in Africa as they're impacted by transgenic potatoes, because they're starting to roll out transgenic potatoes and doing field testing under confined field trials in Uganda. And Ann is here through the end of October, and it may be, I saw your schedule, there might be opportunities to bring her over to talk about transgenic croplands in Africa and what works and what doesn't, and what the fears are, and all sorts of things. So,
please email me about that. I'd love to make that happen.
Okay.
Great. And I'll take 30 seconds. Jason Delborn also dropped a daughter off at college at UNC, so much easier. I went to Peru last week, invited by the U.S. Embassy and USDA and the Agricultural Innovation Ministry in Peru. I'm working on 4 reports: the IUCN report with Adam and Todd, the American Chestnut workshop report with my team, including Katie and Adam and Dalton, the DARPA Safe Genes landscape analysis of the gene-drive mice, and also the National Academy's Committee on Forest Biotechnology. So I am Mr. Report Man. I don't advise you to work on forest events. Next week, we have Paul Vincelli. Is it Vincelli or Vincelli? Do you know?
We'll find out.
We'll find out. It's Vincelli. And the title of his talk is "Can CRISPR Displace Crop Pesticides?" I know that he will be here and is open to visits, so I'm taking him to breakfast on Wednesday. I know Jennifer has an appointment with him, but he has space in his schedule, so please get in touch with Patty and Sharon, since Sharon's out of the office.
She'll be out till next week, so just get in touch with me.
Just with Patty.
And he also co-hosts the podcast Talking Biotech.
That's
so
right.
if you ever get, and Jennifer and Fred have both been on his podcast, which is posted on our website. So if you, so if you want to share any of his past stuff, look at our website. We've got it there, especially his interviews with them.
Awesome. Thank you all for coming. It was good to see you all. I hope you enjoyed that. See you next week.
We can get on our schedule.